Howdy, my name is Hayden Schappell, and this is my Pechu presentation on bacterial evolution, which was a paper on the taxonomy of life by Carl Woese. And so the format of this is Pechu, so it's 20 seconds a slide and a little bit more free form, and so we'll go from there. This is my outline. So I'm going to talk about the paper itself, where things began with biology and how we classified different species. And then we're going to go into how we were able to use RNA. Carl Woese was able to use RNA and discover archaeobacteria, now archaea, and then where things are going to go in the future. So this is the paper itself. This is called Bacterial Evolution. It was in the Microbiological Review in 1987. So Carl Woese, he was at the University of Illinois, and he's really the grandfather of all that we know about sequenced genetics. And so we as humans have this fascination with our past. Where do I come from? Who are my grandparents, my great-great-grandparents? We do genealogy and family history work. We have scrapbooks. We like to keep family stories. We pass down oral traditions. And, this leads to a bigger question, where do we come from before my great-great-grandparents? Do we come from monkeys? There's different theories about that. Do we come from something even older? Something that was single cellular and grew into something that was more complex and eventually monkeys. And so there were kind of three domains that people started classifying different species of life into. So there was the plants, the animals, and then pretty much everything else. And those were your microorganisms. And so specifically, they were called Plantae, Protista, and Animalia. But this was the early form. And so how do we classify those microorganisms then, right? So plants, animals, and then everything else. But within everything else, there's a whole bunch of different stuff. But they really had a hard time. They could only look at them through a normal microscope. The electron microscope didn't come until 1930. And so it was really kind of an exercise in futility. And Carl Woese actually talked about this. And to him, it almost sounded as though he thought biology was at an existential crisis. People weren't going to be able to take it seriously because it was less scientific and more about what we could see. So he says biology's base, scientific materialism, Einstein, Bohr, and Schrodinger could no longer subscribe, basically saying, biology could not keep up with physics. So this is eventually in 1930, they had the electron microscope and they were able to look at the microbacteria even closer and could tell which ones had a nucleus and which ones didn't. So your eukaryotes had the nucleus, the prokaryotes didn't. And Edward Chaton, a French biologist, was able to kind of start classifying things based on genotypes more than just phenotypes. But this didn't really lead a good explanation about evolution. So there were different theories about evolution. Darwin had a warm little pond theory. There was the Aparian ocean, but we couldn't tell where things came from truly. It was just based on theories. And then eventually, we finally could start sequencing RNA. We used RNA, and I'm going to pronounce this wrong, but it's oligonucleotide. Anyway, the nucleotides. They were able to distinguish different phenotypes and genotypes finally, based off of RNA sequencing. This was good, but eventually we were able to do much better using the 16S RNA. This replicated fast enough that we could get good samples and see evolution over time. It was large enough that we could observe it, but it was small enough that it wasn't cumbersome, and it was easily to sequence directly from the enzymes. So there's a couple different methods. I'm not going to go into super detail on each method and necessarily how they did this. But the first method was a distance matrix, and they were able to assign a number based on how far different sequences were from a similar sequence in another species. The second method, they were looking at groups, and it was the smallest number of overall mutations. And so this was a little bit better, but again, this was computationally cumbersome as different genetics would mutate, even if it didn't change the phenotype. And so it would still be the same species. And the third method is the cluster analysis, where we were able to look at things much quicker. Uh, we could look at it was less accurate though, and there were different positions um, where they would find these sequences. Okay, and so Carl Woese was able to look at these sequences of RNA. He would show up to work in his khaki pants and his red flannel shirt, and every day he would look at these images all by himself, trying to find common clusters of um, sequences that matched other species. And what Carl Woese found was that there was something completely different than what we understood before. He discovered archaea. Initially, it was called archaeobacteria, but then we've kind of dropped that bacteria because they're not really a, like bacteria. There's something completely different. But we were able to see how we are all connected, but not connected. And so why does this all matter? Well, if you look at the Earth over time as a clock, a 12-hour clock, humans have only been around for 2 seconds. So we're so insignificant. I got a good quote. Now for this cause, I know that man is nothing, which thing I never had supposed. And so the archaea bacteria, you know, they lent themselves to show that there's much more connection and difference than what we had previously assumed. And this leads us all back to, where do I come from? Well, it's likely that we all come from a common ancestor, Luca, and living things are closer related than we thought. And so where does this lead us into the future? Well, it's possible that we'll be able to find that, last common ancestor, but it's possible that that was only a branch of an even larger tree. And genetics is going to continue to give us this insight, and this is the future study that we can look forward to.